Okay, so here is your final exam review since I ran out of time in class. Both classes went over the introduction, evolution, and basic chemistry in water. Where fifth hour left off, I believe, is on proteins. Be able to characterize the R group on an amino acid. So there's basically two main categories. It can be, if you remember the R group, you got your essential carbon, your hydrogen, we did this in class, your amino group, your carboxyl group, and then your R group. The R can be either nonpolar or polar. So things that you look for it to be nonpolar would be a hydrocarbon, so a bunch of carbons and hydrogens bonded together, kind of like a lipid. If it was polar, maybe you would see a charge, or oxygen and nitrogen sometimes. Those are things that are polar. And so this would make it ionic as well. Nonpolar would be hydrophobic. Okay. The next thing are the levels of proteins. There are 4 levels of proteins. Let me move this up. You have level primary, secondary, tertiary, and quaternary. For primary, remember, that's just the peptide bonds. It's a linear sequence of the amino acids. Your secondary is due to hydrogen bonds, and that can be alpha helix, right, or beta pleated sheet. And then you have tertiary, which is 3D, and that's due to all the bonds, and so that's going to be a 3D shape. And then you have quaternary, which is due to all the bonds, and that's when you have more than 2 interacting. Okay, so there's a question on your levels of proteins. All right, continuing proteins, what affects, I just realized I typed that wrong, spelled it wrong. What affects protein activity would be pH levels, temperature would be another one. Okay. Denaturation occurs, so if you have this pH and this temperature, that's going to change the shape. Whoops. And that is usually irreversible. All right, for nucleic acids, the monomer is a nucleotide. Nucleotides have 3 parts. You have your sugar, your phosphate group, and your nitrogen base. And that's the basic DNA structure. The nitrogen bases that are in DNA are going to be adenine, guanine, thymine, and cytosine. And for RNA, it's going to be adenine, guanine, uracil, and cytosine. So we'll get into this more second semester. The bond for a nucleic acid is called phosphodiester bond. All right, moving on to cells. All right, smaller cells are more efficient because they have a higher surface area to volume ratio, which means they can get their food in enough time and get rid of their waste more efficiently. Eukaryote versus prokaryote cells, you need to know that eukaryote cells have organelles, have a nucleus. Prokaryote cells have no nucleus, no organelles. Examples of a eukaryote cell would be plant, animal, fungi, etc. Prokaryote are bacteria. That's your only example for a prokaryotic cell. Peroxisomes detoxify cells, gets rid of poisons, drugs. Also a poison that's made in metabolism, hydrogen peroxide. So it gets rid of the hydrogen peroxide in our body. A lysosome is similar. It breaks down old organelles. It has digestive enzymes to do that. Okay. And that's it for that one. Okay, for vacuole, plant vacuoles in particular have many, many functions. They help maintain the trigger pressure to help that plant stay upright. Another function that a central vacuole will do is storage of water, which we talked about in class. It also stores protein, so it stores water. It can store protein in there, enzymes in there, waste in there, all kinds of stuff. Another one that it does is it can do lysosome activity. Because plants do not have lysosomes, plant vacuoles do that job of breaking down the organelles and any unneeded cellular materials. A ribosome are the dots. They are mainly protein synthesis. A nucleus is the control center of the cell. Controls everything in the cell. The structure of the chloroplast, you have your thylakoids, and the stack of those is the grana, and the inside fluid is the stroma. The function of a chloroplast is to do photosynthesis and extract the light from the sun. You would also want to know where ATP, the formation of ATP is occurring. And if you think about your bubble map, ATP is made on the first electron transport chain, which is occurring on the thylakoid. All right, and then the last one is plasmodesmata, and those are channels in between plant cells. So if you got plant cells, it's a little hole right there, and it allows things to go from one plant cell to the other plant cell. Okay, moving on to membranes and cell transport. Okay, there are lots of different proteins that are on the membrane, if you remember. And we learned about peripheral proteins and integral proteins. And one of the integral proteins that we learned was a transmembrane protein. And these ones extend all the way through the protein. Another protein we learned about that's on the outside of the cell membrane is a glycoprotein. Glyco means carbohydrate. So it's a carbohydrate attached to the protein. Okay. In the Final Jeopardy, we did permeability. So remember, things that are easily permeable through the membrane are going to be small, and definitely nonpolar or hydrophobic. Those things can go easily through the membrane without any problem. All right, then you have your three conditions of osmosis: hypertonic, hypotonic, and isotonic. For hypertonic, hyper refers to high solute outside the cell. So if there's high solute outside the cell, that means there's lots of water inside the cell in comparison to outside the cell. So water is going to leave the cell and the cell is going to shrink. If it's a plant cell, the cell wall will not change. The vacuole will just get smaller and the organelles will shrink down. And it would lose turgor pressure and wilt. Hypo means high. Hypo means low. Low solute outside the cell. So if there's low solute outside the cell, that means there must be a lot of water outside the cell. And so that water will move into the cell and the cell will get bigger. It will grow. The cell wall will not change on a plant cell. The vacuole will just get really full and have a lot of turgor pressure, and the plant will stand upright. An animal cell could possibly burst. Isotonic, everything is in equilibrium. Solute and water, everything is equal on both sides. For the sodium-potassium pump, you have to know numbers and directions. You have 3 sodiums that go out. These are ions. 2 potassium ions that go in, and this requires 1 molecule of ATP. This is a form of active transport. Facilitated diffusion is a form of passive transport, and that is when you have a protein in there and things are going through it. This is the reason why they need facilitated diffusion, is because they're not small and they're not nonpolar. They're too big to go through naturally. They don't have the right permeability, so they need a protein to assist them, and so that is facilitated diffusion. All right, pinocytosis is when liquids are going through the membrane into a vesicle and then into the cell. Okay, for the last two, co-transport is when two substances are going into the cell at the same time or out of the cell at the same time. Exocytosis is getting rid of material. Remember, it's in the vacuole or the vesicle. It goes to the membrane, which is right here, and then the vacuole fuses with that membrane and that material leaves out. All right, starting enzymes. Catabolic versus anabolic. All right, catabolic versus anabolic reactions. Catabolic breaks polymers into monomers. Anabolic takes monomers and turns them into polymers, so it's the opposite. An easy way to remember that, if you remember, is cats have claws, and that breaks and scratches and breaks your skin and furniture and stuff. So catabolic reactions are breaking down large organic molecules into its simpler form. Anabolic are putting them together. You can think of that as an example. Catabolic reactions are breaking down things like in cellular respiration. And in anabolic, it's building it, like when it builds glucose. Okay, the next two terms are opposites, endergonic reactions and exergonic reactions. Endergonic reactions is when energy is absorbed. So you're gaining energy. So that means your free energy, which is abbreviated as delta G, or G, is going to be positive. And exergonic reactions, energy is released, so your free energy goes lower, so your G is going to be negative. Okay, the next two, enthalpy is a measure of the heat in a reaction or in a system, and that is abbreviated H. And then you have entropy, which is a measure of the order of a system, and that is represented by S. Okay, moving on, finishing up enzymes. Activation energy is the energy that is required to start a reaction. Okay, activation energy is going to be lower if there is an enzyme present. Free energy is represented by the letter G. So it just represents the amount of energy that is available to do work. All right, next one you have oxidation versus reduction reactions. This is called redox. Oxidation is loss of an electron. Reduction is gain of electron. Oil rig. All right, for an enzyme you have an active site. So if the enzyme has a specific shape, right, because they're specific, whatever that shape is, that is the active site, and that is where the substrate will bond to in a typical situation. Okay, the substrate is just another name for the reactant in the experiment. Hold on, I gotta let my dog out. Okay, the allosteric site. So if you have an enzyme, where this groove is, that's the active site. Another site where something can bond to is the allosteric site. And so when something bonds to that allosteric site right there, that's going to change the shape of the active site, and so that the substrate cannot bond anymore. And that blocks it. And then the enzyme works at its fastest from our lab at the beginning of the reaction because there is more substrate available. So the enzyme works faster. Okay, starting cellular respiration, this is more recent stuff. Chemiosmosis is the pumping of the protons through the proteins, right? And that creates a proton gradient, and then those protons get pumped back through ATP synthase to make ATP. This happens in both cellular respiration and in photosynthesis. Decarboxylation is the removal of a carboxyl group. So the carbon dioxide gets released, and that hydrogen usually will add to an NAD to form an NADH. Dehydrogenation is the removal of a hydrogen, which can also make NADH. And then you should know your bubble map. You need to know the ins and the outs of all four stages, just like you did for the test. Hold on, my dog wants back in. You also should know the locations of all stages. Remember, stage number 1 requires glucose. It breaks down glucose. It needs 2 ATPs to break down the glucose. You end up getting NADH and ATP made, a net of 2 ATPs, and that is done in the cytoplasm. Stage number 2 is the formation of acetyl coenzyme A. It occurs in the matrix. It requires pyruvate, which you made in glycolysis, and a decarboxylation is occurring, so you're creating carbon dioxide, and you're making NADH. Stage 3 is the Krebs cycle. It occurs in the matrix. You have multiple reactions going on, but you're making a couple carbon dioxides from decarboxylation. You're making NADHs, FADH2s. You're making 1 ATP. And then your 4th stage is the electron transport chain, and that's occurring in the cristae, and that is requiring NADH and FADH2. You got the chemiosmosis occurring and the electron transport chain. You're making ATP through that proton gradient and ATP synthase, and you are also creating water. Okay, for photosynthesis, you need to know there's a question on the electromagnetic spectrum order from a shorter wavelength to the longer wavelengths. You have this in your notes, but in the Jeopardy that we played, the shortest wavelength is gamma ray. Okay? And then you have your X-rays and your UV light and so on. So you need to know that from short to long in order. It's multiple choice. And then the same thing for photosynthesis. You need to know the 2 stages of photosynthesis, what's going in, what's going out. So you got your light-dependent reactions, which you have water splitting, right? When the light hits the leaf at P680, you have that electron being excited, goes to the electron transport chain. It goes through the 3 different protein complexes with, I'm going blank, cytoquinone, cytochrome complex, and cyanin, plastocyanin. I said that first one wrong. And you're making ATP. And then the tired electron goes to P700, where it goes to all these different electron acceptors, where the last one is ferredoxin, and then we are making NADPH. That is occurring on the thylakoid. And then you have your Kelvin cycle, where you got carbon dioxide joining with RUBP using Rubisco to make our PGA, and then PGA is turned into G3P, which can make our glucose, or G3P can make RP and then regenerate our RUBP. Okay, so the electrons in photosynthesis come from water. There is a question on the parts of a leaf, so please review your parts of a leaf. You need to know where the chloroplasts are in a leaf, so please look that up. And then you have to know the similarities of what photosynthesis and cellular respiration have in common. So think about, do both of them make ATP? Do both of them make NADH? Is NADPH in both of them? Is chemiosmosis in both of them? Is electron transport chain in both of them? What about carbon dioxide? Is that similar? What about oxygen? Is that similar? So you want to look at the similarities between photosynthesis and cellular respiration. It's a multiple choice question. Okay, so when you're doing your short answer, your first problem is going to be a surface area to volume ratio problem, which we've done in the past. But let's go over it. The only difference is going to be that number is going to be different. Okay, so this is our practice problem. We're doing a cube, and it will still be a cube when you do it on your exam. So if you look on your formula sheet, which you'll have, you need to calculate 2 things. You need to calculate the surface area of a cube, and you need to calculate the volume of a cube. The surface area of a cube on your formula sheet is going to say 6a squared, and the volume of a cube is going to say length times width times height, which is the same thing as a cubed. All right? So first you need to know and understand that a is the side of the cube. Okay? So an a in this case is R2. So for surface area, that's going to be 6 times 2 squared. So that is 6 times 4, which is 24. And our a cubed is going to be 2 cubed, which is 8. So this is like a surface area over volume. So you're going to put 24 over 8, which is 3. So your answer is going to be 3. And that would be centimeters cubed, or squared, sorry. And that is how to do surface area to volume. Let's move on to solute potential. From your formula sheet, you will remember that the solute potential equals negative I CRT. Okay, so from the problem, you need to figure that out. All right, so we have negative. What is I? I is ionization constant. It has to do with what the solution is that's inside your beaker or cup or whatever you have. And in this case, if you read the problem, it is NaCl, which is a salt. Salt ionizes into sodium and chlorine so that you have 2 ions. So your answer would be 2 for I. Your C is the molar concentration. It's given in the problem as 2.7. Okay. R is always the same. It's going to be on your formula sheet. If you don't remember, it's 0.0831. And then you multiply that by temperature, which is 35 degrees Celsius, which you need to convert to Kelvin. All right, so do that. We're going to take our degrees Celsius and add 273 to it. So 35 plus 273 is going to equal 308. So that's what we're multiplying here. All right, so 2 times 2.7 times 0.0831 times 308 is going to equal, that is going to equal negative 138.2, and your unit is going to be bars. And so that is an example. So your problem is going to be very similar to that. And it's an open beaker, by the way. So you don't have to worry about that. Plus, you're calculating solute potential, not water potential. The molarity is probably going to be different. The solution might be salt. It might be sugar. If it's sugar, like glucose or sucrose or whatever, then your ionization constant would not be 2. If it's sugar, it's going to be 1. And then your temperature might be different. So it's the same steps, though, as this. Okay, for labeling a cell, it's going to be similar to the one that's in your review. I don't have it on the on here, so you're just going to have to listen. But you're labeling most of the letters there, okay? And it's not going to be that exact diagram, but it'll be a very similar diagram. This one is an animal cell. I believe you are doing a plant cell on your final exam. So practicing on the animal cell, A, you should have got Golgi body. B is pointing to the lines inside the cytoplasm. Those are called cytoskeleton. C is a smooth ER. I know it looks a little funny, but there's nothing else it could be. It's a smooth ER. D is pointing to the nuclear membrane or nuclear envelope. E is pointing to the nucleolus. F is the nucleus. G is cytoplasm. H is rough ER. I is mitochondria. J are pointing to the dots. That's ribosomes. So then you look at K and you think, what is that? Well, there are probably multiple answers for that because you're not sure. It could be a vacuole because animals have many small vacuoles. It could be a lysosome. Who knows? It doesn't look as small, but it possibly, looking at where the Golgi body is, it could be a vesicle. My guess is it's a vacuole, but you won't have anything like that probably for the plant cell diagram, most likely. L is centriole, and then M is your cell membrane. All right, so lastly is our Hardy-Weinberg problem. And so let's go through this. It says 20% of a population has blonde. Okay, so for Hardy-Weinberg, that tells you that 20% is blonde hair. And so that's a person that has blonde hair. So 20% is represented by 0.20. Oops. And so obviously if that's a person, that's not an allele, so that equals Q squared. I guess that's the hardest part. Once you have Q squared, you can get Q by taking the square root of 0.20. And so if you do that in the calculator, you're going to get 0.44. And then using your formula sheet, you know that P plus Q equals 1. So if you subtract from 1, you get your P, which is going to be 0.56. So right away, we already answered the first question. Calculate the allele frequency. The allele frequency is P and Q. All right, second thing you need to do is genotype. You have 3 genotypes. It can be homozygous, heterozygous, or recessive. So homozygous, dominant, is P squared. So you can easily figure that out. To do heterozygote, it's 2PQ. You can plug and chug that. And you already did your Q squared right there. And so then you need phenotype frequencies. So there are 2 frequencies, blonde hair and I'm assuming it's going to be brown hair. And they told you 20%, so the frequency of blonde hair is 0.20. And so obviously the frequency for brown hair would be 0.80 because it has to equal 1. And so you're going to have to do all 3 of those for your problem on your final. Okay, so I told you there were 5 short answer problems, and there are only 4 on your review because one of your short answer problems are already in the review above. So it's just a short answer, tiny little question. And so then, as I talked about in class, you have 2 essays to do. First time you have 2 essays to do. You're going to actually have 3 choices, and you need to do 2 of the essays. All right, so let me write this out. So you're going to have 50 multiple choice. You have 5 short answer. And then you're going to have 3 choices. So your choices are going to be, choice number 1 is going to be a Hardy-Weinberg essay. Okay. Choice number 2 is going to be on cellular respiration. And choice 3 is going to be on macromolecules. So you're going to need to be comfortable with 2 of those topics, and the one that you're not comfortable with the most is the one you skip, and you have to do the other 2. So half of your grade for your final exam is going to be on the 50 multiple choice, and the other half of your grade is going to be based on the 5 short answer and your 2 essays that you do. Okay? And obviously the curve will be there. So good luck. If you have any questions studying, you can email me. Thanks.